a single C allele, you're 3 centimeters more. If you're dominant for L, you get 40 centimeters. Straight up. So now let's have a plant. What will be the height of a plant that has the genotype big A, big A, big B, big B, big C, big C, big L, big L? As long as you have big L, you get 40 centimeters. Doesn't matter if you have 2 copies of big L or 1 copy of big L. You're going to get 40 centimeters. There's your 40. And then I said each allele of the A, B, and C gene adds 3 centimeters. So since you're homozygous AA, you'll be 3 plus 3. Homozygous big B, 3 plus 3. Homozygous big C, 3 plus 3. So this is an additive effect. So the total height of the plant will basically be, in effect, whatever that is. Okay, now you could have a plant that's little a, little b, little c, little c, big L, big L. This will be 0 plus 0 plus 0 plus 0 plus 0 plus 0 plus 4. So it will be a 40 centimeter. And then it can get complicated. You can have a plant that, what would be the height of the offspring produced from a cross between plants A and plant B? So this offspring will be heterozygous. Big A, little a, big B, little b, big C, little c, big L, big L. Correct? And so it will be 3 plus 0 plus 3 plus 0 plus 3 plus 0 plus 4. So the offspring will be an intermediate between the 2 parents. So it will look like an incompletely dominant trait. What would be the height of an offspring produced from a cross between this heterozygous and this heterozygous? So you basically have to draw out all of them and basically figure it out. You just have to add the leaves. And that's what you see when you get things like genes for hair color and skin pigmentation. These are additive effects of dominant alleles in a trait. So for example, skin color, you have 3 different genes. And when you are recessive for all the alleles for those 3 genes, you're light skin. If you're dominant for all, so you have 6 different alleles, 2 for each gene, you are dark skin. And you have a whole gradation. Similarly for hair color, I believe there are 5 genes that are involved. So multiple genes affect traits. And so hair color is controlled by multiple genes, 4 genes, on chromosomes 3, 6, 10, and 18. Okay? The more dominant alleles that appear in the genotype, the darker the hair. So these are known as polygene traits, i.e. multiple genes affecting a single gene. And sometimes they're additive, sometimes they have other rules. We are doing simple genetics, so we're talking only about additive effects here. Yes?
Is this similar to like incomplete dominance or more like fast level?
In a sense, incomplete dominance, but it's incomplete dominance is for a single gene, right? But it's in a sense that's what we're aiming for. But for multiple genes. So what happens is, and that's exactly what this is. Here is your incomplete dominance, right? 2 different phenotypes underneath each gene. That's heterozygous. But when you have 2 genes and you have additive effects, you get different phenotypes, right? And so here is your 1:2:1 ratio for your classic incomplete dominance. But then when you have 2 genes, you notice this broadens because the number of classes increases and the height decreases. And it's this pattern that you basically are measuring to figure out the number of genes that are playing. In the very simplest sense, we're looking at this kind of changes in pattern to determine and tell you how many genes are playing a role in the trait in this study, in a population of individuals. That's how you basically do it. So these are continuously varying traits. They're also called quantitative traits. And in the literature, you will see things that are called QTLs, quantitative trait loci. So for things like Parkinson's and Alzheimer's, you'll read papers and they'll say, what QTL analysis was performed? So basically analyzing things like this in a population. And they're trying to figure out how many genes are involved, how many alleles of each gene is playing a role. And so, you get these curves. And the curves are dependent on the number of positive dominant alleles. So in skin pigmentation, 3 genes, big A, big B, and big C. And you can have 1 copy of big A or 2 copies of big A, 1 copy of big B or 2 copies of big B, 1 copy of big C or 2 copies of big C. Here is what they would look like. Because there are 3 genes, not 2 genes. So you have 8 classes of gametes. And then you combine all 8 classes and you get the patterns going from darkest to lightest. And each class will have a certain number. And you can basically figure it out. Now, I'm showing you this. These kinds of questions are really hard to give you as homework off of exam. But I think you need to know about it because most of the genetics that we study today is focused on quantitative bits. The easy bits, low-hanging fruit, we've collected. Phenotypes that are involved that have a single gene mutation. We know about those already. It's these ones that are now the focus of most research. How is diabetes caused? How many genes are involved in it? Heart attacks, coronaries. Why are certain individuals more prone to atherosclerosis and cholesterol plaques in their blood? All of these are caused by multiple genes affecting the same phenotype. And so most medical research today is focused on quantitative traits. And so it's important for you guys to at least understand the easiest aspects of quantitative. The mathematics is very complicated. Usually you have computer programs that do the mathematics for you. But you as a researcher have to go into the field if you're a biologist, and you have to interview individuals and you have to basically quantitate or try to quantitate their phenotypes. And so you basically say, okay, this individual is, X brown color, or this individual is, you give them grades. You basically have little color patches that you match against their skin and you give them a number. You go to the next individual in a family, etc., etc. You build these huge charts. Charts get so big very soon that you as an individual can't figure them out. You basically input the data into a computer. There are programs that then ultimately will spew out diagrams and give you ideas about how many genes are involved in this. So, the more genes affecting, the smoother the bell curve, etc., etc. You basically get the picture. Now, in very small brackets, I've also mentioned some traits are affected by the environment. So if you're really researching a topic like this, you really have to pay attention to all of these issues. And the environment can obviously affect how you analyze these. And so it becomes tricky. It's not as easy as I'm making it. But you should be aware of this. Okay. So here is just an example how things spread out and change, and that gives you an idea. Okay. So many multi-gene diseases result from mutations in more than one gene. And a number of traits result from mutations of this. These are all very complex gene traits. All right, Alzheimer's is one, and this is a paper that is a rather famous paper in the Alzheimer's field, where they confirmed, they did a huge analysis. And Alzheimer's, basically, there is a mutation in the gene called apolipoprotein E4. And this mutation predisposes you to Alzheimer's. So I'm sure you've all heard of Jim Watson, correct? Right, Watson and Crick, Jim Watson. He had his DNA sequence. He's rich enough he could afford it at the time. Spent, I think, $1 million on something. I don't know the exact number. He had this DNA sequence, and this is not an urban myth. He insisted he did not want to know the allele for the apolipoprotein E4 gene that he had in his DNA. Because if he had the mutation, he would likely to get Alzheimer's, and he didn't want to know that. But it predisposes you. It increases the probability that you will get Alzheimer's. It doesn't mean you will get Alzheimer's. There are many, many other genes that affect this gene and result in either increasing your probability of getting Alzheimer's or decreasing your probability of getting Alzheimer's. And there was this big study that was performed, and they basically found 27 different genetic interactions in 4 different biochemical pathways that affected Alzheimer's, or a predisposition to Alzheimer's. So it's a very complicated disease. But they're using principles very similar to what I just showed you. So you can basically begin to understand. All right. We're going to skip that. I think we are done with pathways. We're done with pathways. All right, let's take a quick break. I've gone over, it's 9:15, but we'll take a 5